Welcome to the Purdue University Davidson School of Chemical Engineering. This is where we prepare chemical engineers to solve real-world problems, where we challenge them, support them, and give them the experience they need to succeed. Today you'll hear from some of the graduate students and see a snapshot of some of the research happening in the Davidson School of Chemical Engineering. Welcome.
Hello, my name is Jonathan Turnley. I'm a third-year PhD student in Professor Rakesh Agrawal's Solar Energy Research Group in the Davidson School of Chemical Engineering at Purdue University, and today I'm at the PV Electric Experimental Farm. The work that goes on here is a collaboration between researchers in chemical engineering, electrical engineering, agronomy, agricultural and biological engineering, and agricultural economics. And together, we seek to develop sustainable solutions to society's food, energy, and water needs. Specifically at this research site, we're looking at the dual usage of land for both agricultural production and energy production. Over the past decade, solar energy technologies have improved remarkably, both increasing in efficiency and decreasing in cost. However, one big challenge for solar energy is that it takes a lot of land to generate the necessary quantities of energy. The largest consumer of land in the United States is agriculture. So we're seeing the emergence of a future competition over land between solar energy production and agricultural production, unless we can develop some sort of disruptive technology that allows for the integration of solar energy into agriculture. That line of thinking led to the emergence of the PV electric system. As you can see behind me, we're using elevated solar panels, also called photovoltaics or PV panels, which are raised by support structures well above the crops beneath. And by raising the panels, we allow for plenty of space for normal farming operations. This system allows for agricultural production. In this case, we're growing corn, and energy production through the use of the PV panels in the same land, hence the name, the PV electric system. There are still some challenges for this system, in particular because both the solar panels and the crops need sunlight. You can imagine that if the solar panels lead to too much shading on the crops, this could lead to unfortunate yield losses. Researchers in our group are studying this problem and have developed really intricate models to better understand the light distribution within the system. Additionally, we've spent the past 2 summers collecting crop growth data from over 1,500 individual plants. We hope to use this information together to better understand the interrelationships within our system. And from there we want to optimize our system to maximize the total output of both food and energy, and to guide the future of this transformative technology.
Hi, my name is David Rocky, and I'm a 5th year PhD student in Professor Rakesh Agrawal's Solar Energy Research Group in the Davidson School of Chemical Engineering here at Purdue University. Right now, we're in our labs in Forney Hall, where we design, fabricate, and characterize the printable solar cells that are the focus of our research. Being able to print solar cells will speed society's transition to renewable energy because it will enable high-throughput, low-cost manufacturing of high-efficiency solar cells. We envision that one day we'll be able to print solar cells the way that we can print newspapers today. In our lab, we have the ability to take a bare substrate and turn it into a completed solar cell. At each step in this process, there's a wealth of understanding that we've gained on how to synthesize better devices. We've used this knowledge to synthesize a number of different materials that we screen for their merits and drawbacks as we seek to develop high-efficiency, solution-processed, inorganic solar cells. We focus on inorganic solar cells because they're known to be stable and last for upwards of 25 years in the field. To make a solar cell, we start by synthesizing our inks in these glove boxes, which are oxygen and moisture-free environments. We must carefully control the chemistry of our ink so that when we dry and anneal our absorber layers, we can form the desired material without trapping any impurities in the film. We must also control coating conditions to cast our light-absorbing materials, which we can do with a variety of coating techniques, including industrially scalable blade coating and slot die coating, using custom coaters that we created by modifying 3D printers. We also study the heat treatment of the solar cell absorber layer in a furnace such as this tube furnace or this rapid thermal processor. We then deposit subsequent layers onto the absorber layer that allow us to extract electrons from the device so they can be carried to an external circuit. We use a simulated solar spectrum to measure the overall efficiency of the solar cell, in addition to numerous other measurements we can make to characterize the materials properties or the electronic properties of our devices. The success of our work will result in fully solution-processed, printable solar cells. This endeavor sits at the intersection of many fields: chemistry, material science, electrical engineering, and semiconductor physics. In our research, we have the rare opportunity to work in a critically important, deeply interdisciplinary subject area that is slated to grow exponentially in the coming years. If you're interested in learning more about our research or joining our team, please visit engineering.purdue.edu/RARG. Thank you for your time, and I hope you've enjoyed this brief overview of our research.
Hello, my name is Chung Tai, and I'm a fourth-year PhD student working with Dr. Biomedical at the Davidson School of Chemical Engineering at Purdue University. So here I'm going to give you a virtual tour of our research facility using our soft matter research. And soft matter such as biological membranes and particular suspension in complex fluids, they are very commonly seen in biological or industrial applications. And our research utilizes theories, numerical simulation, as well as experiments to understand the fundamental physics behind these complex systems. And these information will be specifically useful in developing a more efficient way for material processing and production. So aside from our main lab, we also have two separate rooms for specific uses. So one of them is the optical room where I'm in right now. In this room we have a multifunctional microscope. We can visualize our micro-sized particles, and also we can conduct our microfluidic experiments over here. And we also have a high-speed camera that can go up to several hundred, even to 1,000 FPS connected to this microscope that can capture high-quality images that allow us to analyze the particle dynamics within our system. Now with this parameter, we can perform different tests on our fluid sample. We can measure the viscosity and even the viscoelastic responses of our fluid sample. And these information, they are especially important for our microfluidic experiments to understand the particle dynamics within our system. So that's it for the tour today. I hope you enjoyed this video, and we're also very willing to answer any questions if you are interested in our research.
Hello, my name is Kyle Wettrack. I am a third-year graduate student in Dr. Chong-Li Yuan's group in the Davidson School of Chemical Engineering. Today, I'll be giving you a tour of some of the lab facilities used in the biochemical and biomolecular engineering research areas. We use these lab facilities in our research to reveal the impacts of epigenetic changes between healthy and diseased cells using our novel single-cell tools. This is our cell culture facility, where we maintain and perform numerous experiments on many different types of cell lines. Here, Han is adding drugs to her neuronal cell line to prepare them for later fluorescence imaging. Now Han is going to take the sample to our high-throughput fluorescent microscope.
Can you help me take some images?
Sure. This machine can image plates up to 384 wells at a time. It features objectives of 4, 10, and 20x, along with 4 different fluorescence filters. It features automatic focusing and scanning the plates, so after setup, we do not need to waste time taking all the images individually. After setting up the plate, Junkai can acquire all his images without even being here. In addition to our tissue culture, we do molecular biology, including cloning, Cas9 editing, and protein engineering in this lab facility.
Hello, my name is Dushyant Vishwanath.
I'm Daniel Tejamar.
And we are third-year PhD students from the group of Professor Yu-Yon Wan in the Davidson School of Chemical Engineering. Our research specializes in the biotechnology research area with a focus on nanomedicine for cancer and respiratory distress. Today we'll be showing you some of the facilities we use in the biotechnology research area.
Our research first starts by synthesizing our polymer materials. In order to do that, we utilize a Schlink line as shown here. The Schlink line allows us to alternate between vacuum and inert nitrogen gas, which creates optimal reaction conditions for the polymer synthesis.
Synthesizing the polymer, the next step is to characterize it. One way to do it is via gel permeation chromatography, or GPC. GPC can provide valuable information regarding the molecular weight of the polymer as well as its polydispersity.
After synthesizing the polymer materials, they can be used for a variety of applications. One of those applications is for nanoparticle formulations. And these formulations can be characterized using dynamic light scattering, or DLS, which is the instrument shown here. DLS allows us to measure the nanoparticle size and suspension. Another tool for characterizing our formulations is what's called a Langmuir trough, as shown here. The Langmuir trough helps us to measure the surface mechanical properties of surfactant formulations by spreading them on the water surface. In addition to measuring the surface mechanical properties, our Langmuir trough is equipped with a Brewster angle microscope, which allows us to visualize the monolayer structure during compression of the monolayer.
A crucial step in our research is to test the efficacy of our nanomedicine on biological models. One way to do this is via cell culture work. Cell culture work is to be done in an extremely sterile environment, such as the biosafety cabinet which we have.
This concludes a tour of some of the equipment we use here in the WAN lab for biotechnology research in the Davidson School of Chemical Engineering.
Hi everyone, my name is Siddhartha Akiraju and I'm a fourth year PhD student here in Badoris Research Group at the Davidson School of Chemical Engineering. Today I'll be your host for the virtual lab tour of Badoris Research Group. Today I'll be showing you some of the laboratory facilities used in the design, synthesis, and application of next-generation macromolecular and soft materials. Here at the Badoris Research Group, we define structure-property performance relationships in soft materials and correlate these molecular-level properties to the myriad of applications of these macromolecular materials in advanced energy, water, and healthcare scenarios. In this room, we synthesize advanced small molecules and polymers and characterize these materials in a myriad of manners, including determining the molecular weights of these polymers. We have 4 hoods for synthesis and purification processes. 3 of these hoods are equipped with slink lines and oil baths for various controllable reaction conditions. Additionally, we have 2 vacuum ovens where we are able to completely dry our polymer samples. After completely drying our polymer samples, we then utilize a size exclusion chromatography system for determining the molecular weights of these polymers. This apparatus is a UV-vis spectrometer and is one of the most commonly used machines in our lab, as it determines the absorption spectra of solution and film samples from the ultraviolet to the visible wavelength regions of the spectrum. In this room, we have 2 inert atmospheric glove boxes for the storage and handling of air-sensitive compounds. In one of these boxes, we have a thermal evaporator to fabricate customized circuits for making electronic devices using our polymers. Finally, in this room, we have a Lakeshore Probe Station for the advanced testing of electrical properties of various electronic devices.
Hi, I'm Laura Wilcox, and I'm a 5th year in Professor Raj Gounder's research group in the Davidson School of Chemical Engineering.
I'm Elizabeth Bickle. I'm a 4th year graduate student co-advised by Professor Raj Gounder and Professor Fabio Rivero. And today we'll be giving you a tour of some of the research labs used by research groups in the Purdue Catalysis Center. In this video, we will show you some of the facilities in Professor Raj Gounder's lab.
Research in these labs includes synthesis, characterization, and testing of heterogeneous catalysts for applications that include fuels and chemicals production with lowered carbon footprint and renewable resources, and automotive pollution abatement and environmental protection. In this lab, we have 2 synthesis ovens that are used for the crystallization of zeolite materials under either rotating or static conditions.
X-ray diffraction allows us to identify the crystalline phase of a material. We use this XRD to determine if crystalline material is present in a sample and to confirm the type of zeolite framework we have synthesized.
We have 3 autochems between the catalysis labs, and this is an example of one of them. They are used for a range of experiments, including temperature-programmed desorptions or temperature-programmed reductions, to quantify active sites with either using a mass spec in this case, or TCD or RGA. In this lab, we have 5 custom-built flow reactors that were built by the first graduate students to join each of the projects. They're used for a range of projects, including methanol dehydration, partial methane oxidation, ethene dimerization, and olefin oligomerization. Each reactor has a custom flow unit that was built to control the gases, as well as a 3-zone furnace where the catalyst in a reactor tube is housed, and finally some type of quantification equipment to analyze the products. So in this case it's a GC. We also have some GCMSs. And some of these units are also automated with LabVIEW to control the gas flows on the custom manifold, as well as the software for GCMSs.
In this video, we will show you some of the facilities in Professor Fabio Rivero's lab. These labs are equipped with flow reactors for various reactions. Here we have a flow reactor for protein oligomerization. These reactors are equipped with gas chromatographs as well as mass specs. We have reactors to study NOx conversion on metal exchange zeolite catalysts, as well as 2 other reactors to study light hydrocarbon conversion that are used by graduate students who are part of our Engineering Research Center, C-STAR. This is one of 2 IRs used for in situ characterization. We can dose a gas and monitor the response of the sample over time and with different temperatures and pressures. One way that we use this is to monitor the species that are present on the catalyst surface after exposure to a reactant gas.
Diffuse reflectance UV visible spectroscopy is a technique that can probe transition metal ions in adsorbed species. It's typically used in combination with other characterization techniques to fully analyze structural properties of a catalyst. On this unit, we have a custom-built flow manifold, so we can flow oxidation or reduction treatments to our samples, as well as a furnace that controls temperature in the in situ praying mantis cell. This custom-built reactor and flow manifold were built to bring to Argonne National Lab in Lamont, Illinois. Typically, a group of grad students will go to Argonne 3 times a year, and we bring metal exchange zeolites to study under oxidation and reducing treatments in situ. And we use Zanes and XAF spectroscopy to be able to determine oxidation states and fractional compositions of our metals on the materials.
The 3Flex is an instrument that allows us to perform nitrogen and argon physisorption measurements. Experiments allow us to measure the micropore and mesopore volume and pore size distribution of catalyst samples. This is important for confirming the crystallinity of zeolite catalysts. This instrument has the capability of simultaneously analyzing 3 samples, allowing us to efficiently characterize samples after synthesis.
Hello, my name is Alex Racknader. I'm entering my 5th year as a PhD student in Dr. Monson's research group in the Davidson School of Chemical Engineering. And I'll be providing a virtual lab tour of the Manufacturing Design Laboratory in the Polymer Composites research area. The MD Lab is uniquely positioned to address the high-rate composites manufacturing bottlenecks that have restricted composites from being widely adopted in the automotive industry. We have a fully flexible manufacturing chain, allowing for any resin or fiber to be converted via selected preforms to custom designed parts. Our research focuses on integrating next-generation composites manufacturing approaches with a full-scale Industry 4.0 digital manufacturing testbed. I will show you 2 process lines that highlight our ability to be a turnkey operation for advanced thermoplastic composites manufacturing. First, we make thermoplastic prepreg tape, where we take any dry fibers. Shown here are Kevlar fibers. These fibers are sent to the fixed bar spreader, where the fibers are collimated and form a web with uniform thickness, where it is sent into the impregnation die. This die is fed with molten thermoplastic resin, which is metered by a melt pump. The resin is pumped into the fiber bed using the wave plate design. Here you can see the prepreg tape exiting the impregnation die. The tape is sent through a series of consolidation rollers to compact and cool the tape. The whole line is driven by a belt puller. The tape is wound onto a roll for use in other processes in the MD lab. Here is a finished roll of material. The prepreg tape is used elsewhere in the MD lab. One such example is the Keytech cell, which creates composite preforms for hybrid molded parts. The prepreg tape is sent into an IR oven to consolidate the tapes into a single toe. The toe is formed into a skeleton structure which is capable of efficiently distributing the loads in the final part. The forming process utilizes a 3-axis head and 3-axis table to form complex 3D preforms. This process is fully automated. A robot picks up the preform from the jig and places a new set of bushings for the next cycle. The preform is transferred over to an injection molder for hybrid overmolding. This breadth of equipment and automation allows the MD lab to be a fully functional composites testbed that can start with any dry fiber or resin and produce a custom designed part. Once the hybrid molding cycle finishes, the robot removes the final part and places a new preform for the next cycle. The hybrid molded part is scanned to check for dimensional stability, shown here. And here you can see a side-by-side example of the preform and hybrid molded part.